to you guys? Okay. I know we had a test since then, but anybody had any questions over the stuff we originally talked about? My guess, probably not. It's been pretty much kind of anatomy, straightforward. Thank you. Yeah. Okay. So the ventricles, if we've talked about them or if we haven't, they're chambers in the heart or the brain. So those of you that had lab on Tuesday, you got to see some of those. All right, and usually when they get listed on here, they're always in order of how the cerebral spinal fluid flows because that comes into play. So they're basically cavities or pockets within the brain that's going to be filled with cerebral spinal fluid. So the cerebral spinal fluid washes inside the brain through the ventricles and then out around the brain on the outside, kind of like it did with the spinal cord. It washed around the spinal cord and then down that tube that went down the center of the spinal cord that we called the central canal. Okay. So first one in the brain are the lateral ventricles. I'll show you a picture where these are at. We have a third ventricle, a cerebral aqueduct, a fourth ventricle, and then lastly that central canal. Now, these ventricles tend to get lined with a certain type of neuroglial cell that help move the cerebral spinal fluid. What cell was that? What would that be? Ependymal cells. Thank you. Okay, so lined with ependymal cells. And the other thing we find in this, in and around the lateral ventricles, third ventricles, what's called the choroid plexus. And it's one of the areas that we produce the cerebral spinal fluid. Because really, cerebral spinal fluid isn't that different from like blood plasma. So we take the blood, we run it through this capillary, and think about running it through a strainer. We keep all the proteins, all the cells inside the capillaries, and that blood plasma comes across, and that's what's going to be our cerebral spinal fluid. So this choroid plexus of capillaries is one of the areas of which we produce cerebral spinal fluid. Here's that picture. So here's a side view of it, and here's a straight A to P view of it, anterior to posterior. So these things here are the lateral ventricles. Okay, so they're here and here, and they run one in one cerebral hemisphere, one in the other. Then they connect to the third ventricle right here, and then they run down another pathway which is called the cerebral aqueduct from here to here, and then into the fourth ventricle between the cerebellum and the brainstem. Okay. Cerebral spinal fluid itself, like I just said a second ago, is very similar to blood plasma. Okay. It's got kind of a salty, sweet taste to it. You're thinking, what? Huh? Well, so this cerebral spinal fluid fills the ventricles, the canals. It also flows through that subarachnoid space. And the brain produces about 500 milliliters, or half a liter a day. Okay? Now, it also absorbs a half a liter a day, which means the amount of it stays constant. Okay? We get rid of a half a liter, we produce a half a liter. And that's important because this cerebral spinal fluid helps protect the brain. It rinses away wastes, but it also makes almost like a water fluid, not water, because it's not water, but a fluid barrier for it so it doesn't bang around in the cranial cavity. Okay? It doesn't float on it. It doesn't sink to the bottom. It's suspended in it. Okay? And so sometimes, do they ever look at cerebral spinal fluid? They do, right? They're worried about maybe meningitis. They'll do a LP, a lumbar puncture, and they'll draw out cerebral spinal fluid. Anybody like a CNA right now? Maybe no. Okay. Do you know what the orders are for somebody when they do an LP, when they send them home? They're supposed to stay off their feet for about 12 to 24 hours. That's because now they don't have as much cerebral spinal fluid and their brain isn't protected as well, and they don't want anything damaging the brain. Okay? So the brain's at a little bit of a disadvantage, so they basically want them off their feet until the brain replaces that cerebral spinal fluid that they took off. About 40% is formed in the subarachnoid space, 30% by ependymal cells, and another 30% through that choroid plexus. I'm not going to hold you to those percentages. Just know that those are the three areas that the cerebrospinal fluid's produced. And its function, they'll call it buoyancy, meaning it supports the weight. That's the part where it doesn't float on it. It doesn't sink to the bottom, because if we had our brain and the brain was more dense than the cerebrospinal fluid, it would sink to the bottom. So then all the structures on the bottom of the brain would be bouncing off like those sphenoid bones, and that wouldn't be good for it. We don't want it floating in the cerebrospinal fluid, because then all the top layers of the brain, the cortex, are going to be banging up against the top of, say, the parietal bones. Okay? We want it to be the same density so that it's suspended in it. So if it was an aquarium, it would sink down to about the middle and just sit there and float. Okay? So then it's got fluid all around it to protect it. Okay? Another function, just protection, and then chemical stability. The cerebrospinal fluid will rinse away some of the waste products that the brain produces. The flow of the cerebrospinal fluid. If we produce it in the choroid plexus and the lateral ventricles, it flows from the lateral ventricle to the third ventricle, to the cerebral aqueduct, to the fourth ventricle. Okay? And then now it can go down the central canal or into the subarachnoid space. Okay? Kind of gets a choice. Some of it will go down the central canal, some will go out at that subarachnoid space. And then eventually, it's going to be absorbed back into the venous system at what's called an arachnoid villi, or villus. And think of it as just a little bit of like a valve that lets this fluid flow out, that was cerebrospinal fluid, back into the blood as the plasma. Okay? I think somewhere we'll have a picture of that coming up. So, here a picture again. Lateral ventricle flows into the third ventricle, cerebral aqueduct into the fourth ventricle, and then the central canal is one of the ways it can go. Here's more like the view that you guys had of the brain on Tuesday. Lateral ventricle was in here underneath this white structure. What was that? Corpus callosum. Okay, so lateral ventricle flows this way into the area of the third ventricle, down the cerebral aqueduct to the fourth ventricle, and then it can go out what's called this median aperture and go out around the brain, or it can go down the central canal. Here is one of those arachnoid villus, or arachnoid villi. Okay, so back to this picture, they've taken a little chunk out. We've got bone. What layer would this be? Yeah, it's dura mater, but in the brain we have two layers of dura. That's the periosteal layer, and this layer would be the meningeal layer. And so in between the two layers is where we have some of those dural venous sinuses. So this is a vein, okay, essentially. And this villus pops up here and lets the cerebrospinal fluid flow from here back into the blood. So our other layers, this black space here is the subdural space. This is the arachnoid matter, and this is the subarachnoid space where you find cerebrospinal fluid. And then the layer that adheres to the brain is the pia mater. Okay? Blood in the brain. So, the brain makes up about 2% of your body weight, but it receives about 15% of the body's blood. And so what that tells you is that it's very active, metabolically speaking. It requires lots of blood, which brings oxygen and glucose, right? So it needs a lot of that, which makes sense. Your brain's making all kinds of decisions even when you're not. It'll consume 20% of the body's oxygen and glucose. And it's doing that oxygen and glucose. We use those to make what? ATP. Okay, we need that ATP to set up those resting membrane potentials, right? So all those neurons can fire when they're stimulated if necessary. You'll lose consciousness within 10 seconds if blood flow is interrupted to the brain, okay? Body will shut down. I put you in my triangle choke, you're going out, you're tapping. 10 seconds. Unless you start punching me in the face. Then I'll say, stop that. All right? So, the brain goes without oxygen for 1 to 2 minutes, greatly impairs the neuron function, but you can come back from that. It's when it gets above greater than 4 minutes that it's going to cause irreversible brain damage and possibly death. Okay? So that's usually why when the EMTs race up to the crazy parent's house that's let their 2-year-old fall in the pool while they're smoking their bong or whatever, they ask them how long the baby's been in the pool. Because if the mom's like, oh, like 15 minutes, there's no doing CPR. There's no bringing the baby back. Okay. So blood-brain barrier, you may have heard of that because lots of medications can't cross the blood-brain barrier. The brain, or the body, has this barrier made up of these astrocytes with their little feet that seal off the blood vessels, so we try to really limit what can get from our blood supply to our brain in order to protect it. Okay? Normally, capillaries have little holes in them, and they're designed to exchange nutrients between the blood and any cell in the body. But in the brain, we limit that. This is called the blood-brain barrier. Okay? So, the blood vessel layer itself, it has a single endothelial layer, usually has little holes in it, and then those astrocytes with their little perivascular feet wrap all around it and join real tight so that things can't make it across that normally would. Okay, and that's done by the astrocytes. Now, things that are permeable, because now it has to go, it doesn't have to go out one of the holes, it has to go through the cells of the astrocytes, through their perivascular feet. And often lipid-soluble molecules can do that. So alcohol can, otherwise you wouldn't be able to get messed up on the weekends, and then what fun would there be? Think of how many bad decisions you would have avoided in your life. Okay. Nicotine and some anesthetics can all cross fairly easy. Small molecules like gases and glucose can still cross, but larger molecules, because now those feet have closed down the pores so much, proteins, antibiotics, and several neurotransmitters cannot make it across. And so that's why you hear about the blood-brain barrier, because a lot of the antibiotics can't. So if somebody has an infection like meningitis, they can only give them certain ones. Those certain ones are the ones that will be able to cross. Other ones that you could give for a upper respiratory tract infection won't cross and won't do them any good. Now, it's saying that there's two layers here that the cells will have to pass through. It'll pass through the endothelial layer, is the single layer of the capillary. So blood vessels generally have 3 layers. When you get down to the capillary, they have 1. It's this endothelial layer. It's simple squamous epithelium. But oftentimes it has pores. If it doesn't, they have to make it through the cells themselves. So it has to pass through that endothelial layer and through the perivascular feet of the astrocytes.
In the brain, the endothelium is type, like
yes.
Joined, and then on top of that,
right.
astrocytes too.
Right. Brain development. Okay, so let's see here. 4th week, your brain is well on its way to developing. Okay, so 4th week, that's 2 weeks after mom might realize she missed her period. The brain's already starting to develop. You'll have what's called a forebrain, a midbrain, and a hindbrain. Really high-tech names there. And then the spinal cord. And it'll look something like this here. Okay? And they have other names, prosencephalon, mesencephalon. We're not going to worry about those. By week 5, this is now split into distinctly this here, and it's going to grow to this. So the telencephalon comes from the forebrain, and so does the diencephalon. So our forebrain splits and gives us a telecephalon and a diencephalon. The midbrain is going to give us a mesencephalon. And the hindbrain is going to split into the metencephalon and myelencephalon. Okay? And then these are all going to develop into certain things, like the medulla oblongata, the pons, the midbrain, the thalamus, and we'll talk about them. They're all coming up. So telecephalon, that's our cerebrum. Okay? Diencephalon is going to be more of our thalamus area, mesencephalon, midbrain, metencephalon, pons, and cerebellum, and myelencephalon is going to be our medulla oblongata. Okay? So brain starts to develop early. That makes sense. Week 4, it's pretty simplistic. By week 5, we're going to have all the regions that we're going to develop. They're just going to grow bigger and more prominent. So we're going to start off with the myelencephalon, which was the bottom one, right? We go back to this. Telencephalon was the orange. Myelencephalon is the green. So we're going to start here and work our way upward. Myelencephalon is going to be the medulla oblongata. Okay. So in the medulla oblongata, we have a pair of ridges known as the pyramids. Okay. Now, that's not super important. The pyramids are what are called corticospinal tracts. So we talked about the spinal cord was like an information highway, both sensory and motor information going up and down. Okay? Well, the spinal cord's right down here. You're coming up through this medulla oblongata. Okay? So there's going to be different what they call tracts. Groups of neurons taking information one direction or the other. This happens to be a corticospinal tract, meaning the tract started in the cortex and goes down to the spinal cord. So information going that direction, would that be sensory information or motor? Motor, okay? So the medulla oblongata has these pyramids, which are predominantly, the pyramids are, not the medulla, motor tracts. We have what's called an olive. We used to identify these things in lab, and we don't anymore, so they're a little bit of a waste to have them in our note packets. This relays information to the cerebellum. Okay, so information going either direction would run through this olive and get relayed back to the cerebellum, which doesn't mean anything to us yet what the cerebellum does. The medulla oblongata contains motor nuclei for the cardiac center. Okay, so we have a cardiac center located in the medulla oblongata. What would be your best guess the cardiac center would control? The heart. Good. Anything else? When you think of heart, you think of heart rate, right? You think of anything else? Blood pressure. There we go. So it's probably going to control heart rate and blood pressure. The other thing that affects blood pressure is just the diameter of the blood vessels. So this cardiac center controls some of the blood vessel diameters. Okay? If they get bigger, the blood pressure goes down. If they constrict, the blood pressure will go up. They call that portion of this the vasomotor center. So the cardiac center adjusts rate and force of heartbeat. The vasomotor center adjusts blood vessel diameter. Okay. We also have a respiratory center. So once again, pretty easy to figure out. Respiratory center is going to control the rate and depth of which we breathe. Okay? So two really important things that are handled there by this medulla oblongata. And those of you that had lab on Tuesday, what do we notice about the cranial nerves? If we start higher up on, say, the brain or brainstem, were those the cranial nerves like 10, 11, 12, or those cranial nerves 1 and 2? 1 and 2, and as we went further down the brainstem, the numbers got bigger, right? So if I was going to say there's some cranial nerves associated with the medulla oblongata, what would be your guess what numbers they might be? Yeah, so 11 and 12, that's good, okay? It's actually 9, 10, 11, and 12, okay? So this area, the medulla oblongata, has these reflex centers, and a lot of them have to do with the cranial nerves. So 9 and 10, what did they do? Okay. And I know if this on a test, you'd write them out and you'd figure. Yeah. Vegas? Okay, and so they did swallowing, gagging, some sensory to the back of the throat, okay? 9, 10, 11 was the accessory, right? It did upper trap, sugar, turn head, and 12 was movement of the tongue, okay? So those are the reflexes that have to do with that I'm going to want you to remember. So when we look at this, coughing, sneezing, gagging, swallowing, vomiting, salivation, sweating, movements of the head and tongue, I don't want you to memorize all those, okay? But if you can remember that the medulla oblongata is associated with cranial nerves 9, 10, 11, and 12, they all come off of the medulla oblongata, then those things that kind of go with that, I would expect you to have some idea about, okay? Gagging, swallowing. Gagging can go with vomiting, head movements, tongue movements, okay? Those would kind of all work there. And that's why we learn those silly things partially. So here, and you're not going to see this picture again. This is cranial nerves 9, 10, a little bit, don't teach them anymore, some of 9, 10, 12, I don't know where 11, maybe 11's in there with 12, but that's where they are. Here's the medulla oblongata. Okay. Here you have these bumps, olive, pyramids not shown. So these tracks are running through there. That pyramidal track was a cortical spinal, so it was sending signals down this way. There's other tracks running them up the other way. The olive was the relay station to the cerebellum, so it sent things directly to the cerebellum. Anyway, we don't, like I said, we don't do these cranial nerves anymore. We don't identify any specific structures, so this is a little bit of a waste. Metencephalon, as we come up from the medulla oblongata, what structure do we have? Pons, right? Okay, so the next bump on that brain was the pons. All right. The pons is another pathway to and from the cerebellum. The cerebellum does quite a bit for being just a little portion of the brain back there. And so this pons sends lots of information to and from the cerebellum. Now, we said 9, 10, 11, and 12 were part of that myelencephalon, part of the medulla oblongata. So what cranial nerves might we go with here for this? Yeah, 6, 7, 8. Good. We're actually going to go 5, 6, 7, and 8. Okay, so once again, put on our thinking caps. WTF did they do? Well, 5 did muscles of bite, right? Sensory to the face. 6 did eye movement. 7 did facial movement, anterior taste to the tongue. 8, equilibrium, hearing, balance. Okay, good. So cranial nerves 5, 6, 7, and 8 come off there. Their nuclei are concerned with the things we should know: hearing, balance, taste, eye movements, facial expression, facial sensation. Still does a little swallowing, but some of those that don't quite fit in there. Okay. So these cranial nerves are coming off all in the area of this pons, or between the pons and the medulla anyway. So there's cranial nerve 5. It's always easy because it's big and thick. 5, 6, 7 or 8. So one of these is 7, one's 8. All right. Go up a little bit higher. Or no, stay in the metencephalon. Excuse me. We're not going any higher. We're just going from the pons to the area behind it, which we call the cerebellum. Good. Cerebellum makes up about 10% of the brain's mass. So the brain itself was 2% our body weight. Of that 2%, 10% is the cerebellum, so it's pretty small. But it contains over 50% of all the brain's neurons. Okay? So there's a lot of information being processed there. The inside portion that looks like the veins of a leaf was that arbor vitae. It has specialized cells known as Purkinje cells, which are large globular neurons with lots and lots of dendrites. The more dendrites it has, the more things can synapse on it, the more information it can process. It too also has sulci, gyri, increasing the surface area. What's it do? Muscular coordination is one of the things it does. Okay, so it coordinates all the muscle actions that you have in your body. Okay, which, if you think about it, that's a lot to do a lot of the things you guys do every day and do it with precision, whether that's simply writing, throwing something, typing something, those types of reaching for something without knocking things over. Tactile input and spatial perception. So tactile input is processing the information of things we touch. Spatial perception is depth perception. Okay? So that goes with doing a lot of the muscle coordination things, reaching for something, not reaching too far, not reaching too short. Okay? It does a little bit in the role of passage of time. Okay? I would say the first two are more what it does. But sometimes that's the cerebellum's in the back of the head. Sometimes you hit the back of your head hard enough, and that's when you kind of come to. They're asking you what day it is, whatever, because a little bit of damage there and you kind of lose perspective. Plays a role in speech and emotion. Once again, with the speech, it's a little bit of coordinating the muscles. Motion's not a big thing for motion, but it does a little bit. And interestingly, they found children with ADHD seem to have small cerebellums. And I don't know exactly what correlation they're going to make with that. But may not be able to process everything they need to process. So, going back to muscular coordination. So it's taking, and then depth perception. So take an activity, take shooting free throws, okay, something like that. You're playing some sport, doing something, maybe it's volleyball, whatever, but we say shooting free throws. The cerebellum's where your body's going to take in that input from your eyes on depth perception and then control the muscles that you're going to use to shoot the basketball. Then process, did you make it or not, and make fine-tune adjustments to try to get the outcome you want. And then you practice it over and over and over again so that that becomes more of a very well-used neural pathway that you can call upon all the time. Okay? So it does quite a bit. Going up to the mesencephalon, we're in the midbrain. So in the area of the midbrain, that's where we had that cerebral aqueduct to give us an idea. Those, again, once again, that were on there Tuesday, what was the structure just above the cerebral aqueduct? Inferior. Yeah, superior and inferior colliculi. Good. And so on this picture, this is the superior colliculi, one on each side, and this is the inferior colliculi, one on each side. They call them together the corpora quadrigemina, quad being 4. So beneath here is that cerebral aqueduct running through there, just to give you an idea of where we're at. Okay? It's associated with midbrain cranial nerves 3 and 4. 3 and 4 they deal totally with movement of the eye. Good. We have another thing known as the cerebral peduncles. These are, once again, cortical spinal tracts, motor tracts moving through there. The corpora quadrigemina, which is a term we didn't use in lab, it's talking about the superior colliculi and the inferior colliculi. Now, what do those things do? Well, the superior colliculi does visual tracking. Okay? So we're associated with cranial nerves 3 and 4 eye movements. This corpora quadrigemina helps use your eyes to track something through space. Not only left, right, up, and down, but depth, near, and far. Okay? It also has a visual reflex to help you get your eyes on something. Okay? If you're, let's say, I don't know, you're home alone, or for whatever reason you're maybe you've been watching some scary movie or something, you're home alone, and you walk downstairs. If something like flashes in your field of vision, what do you do? Yeah, your head will jerk around and you'll get your eyes on it. It's obviously to keep you alive, because if it's coming at you, you see it, you have a chance of defending yourself. That snapping your eyes around to it, That's part of this superior colliculi. The inferior colliculi is auditory, but it's the same thing. You come down the stairs, you go in the kitchen, and you hear something behind you. What do? You snap your head around to see what it is once again. But it has to do with an auditory reflex. What your body did was heard that noise and determined where it was in space. It's right effing behind me. You hear a noise that sounds like it's, in the neighbors or way down the road, that doesn't faze you at all. You're like, oh, the neighbors are fighting again or whatever. You hear something right behind you, you're like, what the hell's going on? Okay. Substantia nigra. Now, I want to say that's come up before. Okay, what did that come up with? Yeah, dopamine. So that's the area, so it's in this midbrain area, the substantia nigra. It's where we make dopamine. All right. If we have a problem here, this can lead to Parkinson's disease. So the dopamine was kind of inhibitory, right? And it made the skeletal muscles less likely to fire. Without the dopamine, that inhibition's lost, and what happens is the antagonistic muscles are firing when the prime mover's firing, and they fight against each other. Or sometimes, in this case, that's when they have kind of that shuffling gait. In this case, with the tremors, the muscles are just firing when they shouldn't. So their hand shakes, or they roll the pill, or whatever. And I think I mentioned they usually, if you've ever noticed this with older people, they try to hide this stuff. They'll hold their hands, they'll put something over it just so you can't see, because they're afraid you're going to send them to the doctor and put them in like an old folks home and forget about them. The metencephalon. So back down to the, okay, so medulla, so cerebellum and the pons.
Just the cerebellum.
No, not as much. The brainstem where they come off was where the cranial nerves are. So that would be more with the pons. The cerebellum's kind of like its own structure that comes off. There's no cranial nerves that really come off the cerebellum.
They're still nerves.
Oh, they're still nerves, but not cranial nerves. Okay.
What are cranial nerves? Are they just like more powerful?
They're more powerful. Wouldn't be the word I'd use. They're more important. They're more specialized. So they usually are carrying some sort of special sensation. Okay, whether that's sight or hearing, balance that the rest of the nerves don't carry, or they have to do with like head and eye movement, which allows you to see predators and keep yourself alive, or hopefully keep yourself alive. Okay, then we have what's called the reticular activating system. Now, the fact they're calling it a system, it's not a structure. You couldn't go in and dissect out the reticular activating system, okay? It's more of a group of neurons that run through several areas, the medulla, the pons, the midbrain, that do a specialized job, okay? So this isn't like a thing you're going to find on the brain. It's a system that runs through different parts of the brain, okay? So it spreads throughout the pons, midbrain, medulla. And one of the main things the reticular activated system does, it's very important, it's called habituation. Okay? Habituation is your brain's ability to ignore inconsequential stimuli. Okay? So, think of all the things when you're sitting here in class and you're taking notes. Okay, the person next to you, the person behind you, in front of you, whatever, maybe they're tapping their feet, they're shuffling their papers, somebody walks by in the hall, whatever. And your ability, your brain picks up on all that, but it decides that stuff isn't important and allows you to focus on what you're doing. That's habituation. So think about all the stimuli that comes in, and your brain filters through and decides what's important and what isn't. Okay? And this reticular activating system is, if you can't block out that stimuli, think of how overwhelmed you kind of become. It kind of almost leads to anxiety. That's abilities your brain's ability to block it out. It's that habituation. So that's important. Reticular activating system helps regulate a little bit of balance and posture. It does run through that cardiac and vasomotor center. So habituation is the main thing it does. The second most important thing it does is it regulates sleep and conscious attention. Okay? If you increase stimulation to your reticular activating system, you're more alert. If you decrease stimulation to that center, you become sleepy or tired. Okay? So if you're ever driving at night and you're tired, what do? You roll down the windows, you turn up the radio, any kind of stimulation trying to keep yourself awake. If that stimulation gets too minimal, the brain starts to shut down. Okay? So this is the other, probably second most important thing the reticular activating system does. More stimulation, the more awake you are. It does have some of the analgesic pathways running through there to help regulate pain. But like I said, if I was going to be concerned with the two things the reticular activating system does, it's habituation and regulates sleep and conscious attention. Okay. We go up a little bit higher now, and we're in the diencephalon. The diencephalon is where we're going to find the thalamus. So here on our picture, and this helps people that have lab today, maybe lab will go a little faster. This area right here, this is the thalamus. The area below it, a little funnel shape, that's the hypothalamus. Okay. So the thalamus is described as the gateway or relay station of the brain. It receives almost all sensory information on its way to the cerebral cortex before it gets to the cortex. And this thalamus decides where this information needs to go. Okay? So relay station is probably a better word than gateway. So information comes in, it looks at it, and it decides where to send it. The only sensory information that doesn't pass through the thalamus first is a sense of smell. Okay? Everything else gets thrown through this thalamus. So it processes the information or integrates and directs it to the appropriate area of the cortex. So in a way, it filters things. Probably works a little bit with that RAS for the habituation. What's important, what isn't, where does it go, and how your body deals with it. Okay? And those things all change too, based on kind of state you're in. I don't mean like what state, Arizona you live in, but if there's some reason you might be a little bit scared or nervous, then your brain's going to process, and some of that stuff that it would have ignored, now it's not going to ignore. And you can get yourself worked up into your almost hearing things, or somebody's after me. All right. So the other area up there is the hypothalamus. Okay. So the hypothalamus does a huge amount of things. It's this little area right here, and your hypothalamus is basically the origination of your endocrine system. So you got two governing systems in the body. You got the nervous system, and you got the endocrine system. Okay. We're obviously focused on the nervous system, of which we have the autonomic nervous system, and then we have the rest of the nervous system. The autonomic does everything automatically, right? Well, the other governing system is the endocrine system. So it's using hormones to control things, and it's all involuntary. So this hypothalamus is the origination of those. And it does this by what are called releasing and inhibitory hormones. It communicates with the anterior pituitary, which is right here. So on those brains, we eventually pop off that pituitary so we can see what's underneath. The hypothalamus is what was underneath it. So it secretes hormones down here to the anterior pituitary. The anterior pituitary then secretes hormones, so on and so forth. Those hormones might go down and tell the testes to produce hormones, or the ovaries, or whatever. It also, hypothalamus, controls the autonomic nervous system. So as I just mentioned before, we've got the two things that are controlling everything involuntary in your body, endocrine system and the autonomic nervous system, they're all controlled by this little bitty hypothalamus. Okay? It's kind of crazy. Hypothalamus plays a role in sleep and circadian rhythms, a role in memory. It does a role in thermoregulation. Okay, so when you run a fever, there's an issue in the hypothalamus. The hypothalamus thinks you need to raise your body temperature. Okay, it's doing that to maybe inhibit enzymes of the bacteria or the virus trying to kill it, but it doesn't really feel good to you. It regulates food and water intake and some emotional behaviors. Usually the very basic emotional behaviors like anger and aggression, fear and pleasure. Okay? So quite a lot from a very little area of the brain. Now at the back here, so this was thalamus, this was hypothalamus. At the back usually sits a little thing, they call it the epithalamus or the pineal body or the pineal gland. So here it is back on this picture here. Epithalamus, pineal body, pineal gland. It's an endocrine structure, and it's thought to play a role in the timing of puberty. Okay? So it starts to secrete things that kind of kick-start puberty, which is basically your reproductive system kind of leaving a dormant state and finishing its development. It also, the pineal gland, produces melatonin at night. Have we heard of melatonin? Yeah, so it helps with sleep, right? So melatonin is produced at night. In the absence of UV light, this pineal gland will take serotonin, which we've heard of, right? It's a feel-good hormone or neurotransmitter, and it'll turn it to melatonin at night. Okay? It'll get converted back to serotonin during the day. So there's a correlation, obviously, with good night's sleep, feel better the next day. Good night's sleep, feel better the next day. What happens to people that are depressed? Do they have trouble sleeping at night? Yeah, they don't have trouble getting to sleep. They have trouble staying asleep. They wake up in the middle of the night, can't get back to sleep. Okay? So that's where this melatonin's produced. It's from, anybody remember what amino acids it's made from? Probably not. It's probably more a 202 thing. It's from tryptophan. Heard of tryptophan? What do we eat that has tryptophan? Turkey. So here at Thanksgiving, you eat that turkey, lots of tryptophan. Now you've got more things for your body to use to produce melatonin. Now if we eat the turkey, we get sleepy because we got to watch the god-awful Cowboys on TV. Or the Lions, get the shit kicked out of them. All right? It's horrible. Or Grandpa's story that he's told you 15 times already. You just want to be left alone. Okay, I think that's probably it. So, too much melatonin can make you feel sleepy and depressed, okay? There's what's called seasonal affect disorder, SAD. And those people tend to make too much melatonin. Or seasonal affect disorder is more common for those folks that live where there is no sunshine because they need UV light to break down the melatonin and stop the production of it. So people that live in, say, the Midwest, where in the winter, whether it's cold or not isn't necessarily the point, but it's gray skies. It's always cloudy, that you don't see the sun. Or if you lived in Alaska, where there's parts of the year where you only have 4 hours of sunlight, they all have UV lamps in their homes to get that UV light to try to help them with this type of thing. Okay. So telencephalon are now the lobes of the brain that we've heard of, the occipital lobe, the temporal lobe. They all correspond to the bone that lays over the top of them. So if you learn the bones, you'll know the lobes of the brain. We have a frontal lobe, okay, located behind the frontal bone. And it's responsible for voluntary motor functions. The frontal lobe is a motor lobe, okay? It also plays a role in your motivation, mood, intelligence. Intelligence gets kind of put out into foresight and planning, okay? So if you're able to look off down the road and plan for that, that kind of gets tied into your intelligence, okay? If somebody has a lesion, maybe they have a stroke and an area of that brain dies, it's going to affect motivation, mood, intelligence. It's going to affect their personality, okay? Parietal lobe, this is more a sensory lobe. It takes lots of sensory information and processes it. Now, this sensory information crosses before it gets to the brain. So, if you had a lesion in the left parietal lobe here, that would be a loss of sensation somewhere on the right side of the body, and vice versa. The occipital lobe in the back has where the visual center is. Okay, so sometimes when you hit your head hard enough in the back of your head, you'll see a flash of light. Okay, that's because that visual center is back there. Temporal lobe focuses a little more on hearing, smell, and memory. And then there's another lobe called the insula that's actually been recently discovered in the last 25 years. It's a lobe between here and here. You peel this back and there's another lobe kind of hidden in there. It's called the insula. And it deals a little bit with understanding spoken language and integrating sensory information from inside the body, from visceral receptors. Okay. Any questions on those so far? So it's interesting, they know a lot about the brain because they treated people in mental institutions so bad for so many years. They tried all kinds of experiments on them and stuff like that to find out. So memory, learning, and emotion. So once again, this is going to use what's called the limbic system. We had the reticular activating system. The limbic system isn't so much solid structures, of more regions through other structures. The limbic system has what's called the hippocampus. The hippocampus organizes sensory and cognitive thoughts into memories. Okay? So, but it doesn't store the memories. There's other areas that work on storing the memory, but it kind of forms memories. It also has what's called the cingulate gyrus. It coordinates sensory input with emotions. This is where we form our emotional responses to things, such as pain. Okay? And you've noticed pain's a good one. Everybody kind of responds to pain differently. It's kind of a developed thing, too. As a child, when something hurts, what do? You cry. And part of the reason you cry, one, it hurts, but two, doesn't mom walk over and give you all kinds of attention and whatnot? Okay? So as you get older, you don't, when something hurts, you don't normally cry. Heck, probably more of us, if something hurt bad, we more get pissed off about it than cry, because nobody's going to give a shit. So it's kind of how those things get formed in our brain, how we got shaped. A lot of one's personality is how they responded to something, and was the outcome positive? If it was, that becomes kind of how they handle things, with kind of a learned behavior. If somebody yelled at somebody all the time and got what they wanted, well, then they learned that yelling works. Okay? Anyway, limbic system is another portion. We have what's called the amygdala. Amygdala focuses more on emotions, particularly fear and aversion. So if you have any, what you throw under the classification of phobias, okay, that would be this area of the amygdala. So the hippocampus is more with memory. The amygdala is more with emotions. Okay, does anybody have any phobias in here? I do. I'm afraid of heights. Kind of funny, huh? Yeah, you put me up about more than about 2 stories, 3 stories. It's funny because I used to jump off of houses and stuff. We'd run around in neighborhoods and do crazy shit. But now I can't. Like you put me, I won't even go near the ledge. You start trying to get me over by the ledge, I'll start throwing punches. And the other is claustrophobic. You almost, anybody, well, the C building's all under construction. That's the building way down to south, right off of Thomas. Anybody been in that building before it went under construction? There's an elevator down there. Okay, it's a real small, rickety elevator. So one of the first days they put me in that building, 2 things happened. One, I didn't know at the time. I stepped into the elevator, and it's really small. And there's like 3 students in there. And come to find out later, we have a lot of people on campus that lack common sense. They try to do like what you'd think would be the right thing, but they let some homeless guy like live in that elevator all summer. That thing smelled. So like you step in and you're like, oh, what's that? You know, and you kind of look around and then the door closes. And it's this, like you don't even feel it moving. You're like pushing the button and I'm kind of like, oh, da, 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 da. So I'm trying not to freak out. I don't know if these are my students. And I'm thinking this thing is not only going to just, I'm going to be stuck in this stinky elevator, I'm going to lose my mind. But those are my 2 phobias that drive me crazy. I hate small spaces. I hate being in my office. So it's the other reason I ever tell you guys, if you're coming to see me in my office, let me know, because then I'll make sure that I'm there. Otherwise, I'm wandering around somewhere else. I just hate being in the damn thing. It's too small. Emotions. So the last slide was like the limbic system and what certain areas of the limbic system did. Now we're just taking emotion and we'll touch on some of the areas that all deal with emotion. So the prefrontal cortex, a little bit of how we express emotions. Okay, we said the prefrontal cortex was motor, so this is how we express those emotions. Some people, if they're really happy, might actually cry. Okay, some smile, laugh, whatever. How we express the feelings we have. Some people are really demonstrative. Some are less. Okay, a lesion here would do one of those 2 things. It would either make it over and above what would seem normal, or it would blunt their emotions. Okay. Amygdala, hypothalamus, formation of deeper emotions, and we make associations between aversion and gratification, kind of pleasure and aversion. Once again, they also help shape one's personality. Trying to figure out, do we want to keep going? Show you a couple more slides and we'll be done. So this is the frontal lobe, right? Back here's the parietal lobe, back here's the occipital lobe. Between the frontal lobe and the parietal lobe, there's this central sulcus, kind of divides them, okay? And then we have a pre-central gyrus. Gyrus are those things that stick up, and we have a post-central gyrus. Now we said the frontal lobe, was it sensory or motor? Motor. So the pre-central gyrus, part of that is, because it's part of the frontal, is motor. The post-central is part of the parietal, so it's sensory. Okay? And they've learned, they've mapped out what different areas of this, say, pre-central gyrus, what it controls motorly. Okay? If they stimulated this right here, what that would cause to happen on that person. Or if what they stimulated right here, sensory, what would that person perceive? Okay? And it's a little bit like this. This is the sensory, what they call homunculus. It's like somebody's standing down in this fissure and laying out over the top of it. Okay? So down in here, it's feet, naughty bits obviously, moving on up, head, face, arms, hands, and what you can see by this is the larger a structure represented on this picture, the more sensory input we have for that area. So we have lots of sensory input for the face. Think of some special senses. Think of how important your face is because your brain's behind it. protecting it. Lots of sensory information from our hands, touching things, feeling things, that type of thing. And it's the same thing for the motor. Okay? So the motor was just the precentral gyrus. Somebody standing down in there and laying out over there. And once again, more motor control, fine control for the hands and the face. Okay? And like I said, they learned a lot of this testing on people in insane asylums that were basically abandoned there. They'd drill little holes in their brain, hook electrodes there, stimulate it, and see what happens. Map out brains that way. And see, that's what Grandma is afraid you're going to send her when she has Parkinson's. So don't do that to Grandma. All right, so we'll shut her down there for today.